Hi everyone, I'm Camilla. I'm a genome curator at the GREET team in Sanger Institute. And today I'll talk to you about some of the first insights that we have about the Microbial-Rich sponge metagenomes in the ASG project. So in the Aquatic Symbiosis Genomics project, we are focused on the huge amount of biodiversity that we have in the aquatic ecosystems. So in this project, we provide genetic data for biologists for them to understand how species evolve and live together. And one of these species is a microbe. Don't worry.
There's a thing flashing.
Yeah, the square.
We'll pause your time.
Yes, please. Oh, yeah. Thank you. So really briefly, how the samples are processed in ASG project. So we extract the genetic material, and then this material moves to sequencing and then to assembly. And it's during the assembly stage where the host and the cobiontic genomes, they are separated. So the host usually is a eukaryotic species, so it moves to curation using high-C data. And the cobiontic genomes, they are released as MAGs. And later, these MAGs are submitted to public databases such as EBI Magnify and DNA, and later they are published along with the hosts in genome nodes. So in ASG, we work with many aquatic species. We have marine species, we have freshwater species, but the sponges, they are the ones with the richest microbiota that we have. So this is from where most of our metagenomes come from. And possibly this happens because of the nature of the sponges, because they are natural filter feeders. So if you zoom in a sponge picture, where is the, oh yeah. You can see there are many pores, and the water flows through these pores. And this is how the sponge incorporates most of the cobionts that live inside it. So sometimes the cobionts, they contribute up to 130% of the sponge biomass. So it's really a lot of cobionts. And sometimes the genome curation of the host, the sponge, it's quite difficult, quite challenging because of the great amount of cobionts. So here in the left, we have a high-C map of the sponge Chondrilla caribensis. And this is a high-C map ready for curation. This is the way that we receive it for curation. So for those which are not very familiar with a high-C map, it shows high-C signal that shows the affinity between the chromosomes inside the cell. So we are waiting to see, we're expecting to see the strongest high-C signal only here in the main diagonal. But as you can see, you have some regions which are out of this main diagonal, which possibly are misassemblies that we must correct during curation, or possibly are cobionts that also need to be removed during curation. And we also have here in the red square some really small scaffolds, which are probably from cobionts and need to be removed too. And here in the right, we have a blob toolkit plot. So this plot shows us the distribution of our scaffolds in the assembly based on the packed biocoverage and the GC content. So here we can see we have the bubbles in many colors. So this is many taxonomic groups in there. But if we look at the BTK caption here, we have no porifera. So our main species, our host, our target species, is not tagged by BTK. So actually it's there, but it's masked by the great amount of cobionts. And continuing with the curation stuff, so here we have the two, actually the same high-C map from the previous slides, but in the left it's before curation, in the right after curation. And in this one, we can see that we have two really different regions. We have the green square, which is the sponge genome, only the host, and the red square are the cobionts. And we can see there's a clear difference in the background between these two regions. Here we can see some highlighted regions outside the main diagonal, but that's okay, because that's possibly due to the highly repetitive regions from the sponge telomeres, from the chromosomes, so that's fine. And we cannot see this background here. So these are cobionts that needs to be removed. So us as genome curators, we want to get rid of all metagenomes and all cobionts. Well, we actually don't like them, but so we take them off during curation. And to have a map just like this, it's just the host genome in the left side, in the right side. So in this case, almost more than 50% of the assembly was cobionts and were removed during curation. So what do we have so far in the ASG project about sponge metagenomes? So we have 60 sponge species in the project at the moment, 32 of these species with MAGs, 18 species with the MAGs already submitted, already public. So in total, we have almost 900 metagenomes, where close to 430 are public already. And these are some of the sponge species with the highest number of MAGs that we have in the project. And then we got curious about which microbes are these, which bacteria are these. So if you consider all the sponge species, all the MAGs that we have for sponges, we have a top 5 bacteria there, but the number 1 is the Halococoidea, or Chloroflexi. They are present in 17% of the metagenomes. And this group of bacteria, they are known to transform a wide range of halogenic compounds, usually toxic ones, toxic ones. So they play a really important role in the restoration and conservation of contaminated environments. And usually the Halococoidea group is the most abundant group in the MAGs that we have. An example of this is the sponge species in the bottom, where we have 45% of the MAGs, they are from the Halococoidea. And which would be the main functions of this bacteria, this microbial? So usually they are involved in nitrogen metabolism, especially in deep sea sponges, carbon metabolism, and some of them have some immunological roles, such, for example, microorganism selection. They tell the sponge which bacteria or microbes the sponge should keep or remove, eliminate. And sometimes there is also a crosstalk between the host and the cobiont, trying to protect the sponge as a whole. So, so far, our data showed that. So this is really just insights. This is really preliminary. We haven't done any analysis about this. But what we could see is that then we have the top 4 MAG species, the sponges with the highest number of metagenomes. They are all high microbial abundance sponges, or HMA. And they vary widely geographically, like from Norway to Australia. All of them have a higher number of metagenomes and with many microbial groups. We can see, as expected, the Chloroflexi or the Halococoidea only in the HMA sponges, as expected. And another thing that is expected is a dichotomy between the low microbial and high microbial sponges, which we can also see here, and it's confirmed by our ASG data. So these HMA sponges, they are expected to have more diverse metagenomes, but on the other hand, the microbes are more redundant. So they have the microbiomes are really similar in what concerns the composition and the function, as we can be seen in the deep sea sponge Georgia parva. So this is mostly seen in HMA deep sea sponges. So we believe that what's happening is that we can see a large redundancy of microbes working on the same roles in the deep sea sponges, possibly to bring some instability to the inner system of the sponge. And another thing that might be happening is that this bacteria, or this microbe, they evolve to live, to adapt in a really restrictive environment, such as the deep sea, where they have like less light and a lower temperature and higher concentrations of inorganic compounds, such as nitrogen, for example. So they reduce competition and work as a team to explore the resource they have the most in the deep sea. And the next steps in the project, we have already another 14 sponge species with their metagenomes ready, sequenced and ready to go to public databases. We have new sponges coming to have their metagenome sequenced. And we are now working on the metagenomes of other groups, such as corals, annelids, and mollusks. They will be public soon. And I'd like to thank all my colleagues from Sangha for the great job in ASG, thank the Moore Foundation for the financial support, and thank you all for listening.
Thank you so much, very interesting. I believe we have time for one question. Yep.
Thank you very much. Thank you very much, and fascinating and super quantitative essay and approach, which asks for biological follow-up studies. And you have this. You mentioned in one of the last slides here that stabilizing the inner system of sponges. What do you mean with that? I mean, do I understand that if you remove the microbes, then the sponge morphology collapses? Or
yeah, we don't have this data, right? Because in Sangha, like the curation team, we do like production or research, but possibly our partners in the project, they're studying things like this. But we can see, like I had the help of some sponge people for this presentation, and we can see that the metagenomes, the microbiota, they are essential for the homeostasis of the sponges. For example, possibly with no microbes, they would collapse. Yeah, we believe that.
Thanks a lot.